Hey guys, I'm Tara.
Sierra.
And the enzyme we chose is called thrombin, and it's a bloody cool protease.
Thrombin is a serine protease, which means in its active site there is a nucleophilic serine. Any cut or lesion causing a vascular injury, aka you're bleeding, triggers a cascade response of thrombin. In this cascade, thrombin acts as a primary promoter for coagulation to form blood clots, and it does so by cleaving fibrinogen into fibrin, which it acts as an anchor for platelets in the clotting process. Though it can act as a coagulant, thrombin can also act as an anticoagulant. It does so by being bound to its regulatory cofactor, thrombomodulin, which is depicted as this TM right here in this diagram. In this anticoagulation pathway, thrombin instead cleaves and activates protein C, which acts as a negative regulator of the cascade process, which just means it lowers thrombin's production and activity and degrades it, therefore preventing blood coagulation at inappropriate times. So the inactive form of thrombin is prothrombin, which is synthesized in the liver and released into general circulation in response to any trauma event causing vascular injury. The conversion into the active form is caused by an internal proteolysis, which generates a new N-terminal. It can further be regulated by sodium ions, which we'll get into a little bit more detail later. Once it's activated by sodium ions, thrombin is then converted into its active form, alpha thrombin. This alpha thrombin can take two forms. It can take either a slow form or a fast form, where the fast form is that procoagulation pathway, which is depicted on this right side, which is where you can get your blood clotting, or it can take that slow form, which is the activation of protein C in the anticoagulation path. In that procoagulation pathway, thrombin not only cleaves fibrinogen into fibrin, but it also cleaves factor V, which is going to be really important later on in our presentation when we discuss the paper. Factor V acts as a positive feedback protein, which just activates more prothrombin into thrombin, so you get more coagulation, more clotting.
So now we're going to talk a little bit about thrombin structure. When they crystallized thrombin, they found the catalytic triad, histidine, aspartic acid, and serine, nestled in this canyon-like active site. And they also found exocytes, 1 and 2, that are composed of positively charged residues. And the role of these exocytes is to mediate the recognition of substrates, inhibitors, receptors, foreign objects, what have you. And I'm going to go ahead and link out to our 3D enzyme model through the database Brenda. And right here you can see the active site where the residues are, right, these red guys right here. So that's pretty cool. And as mentioned earlier, sodium has an influence on thrombin, and sodium actually creates a conformational change in the protein. And at physiological pH, thrombin exists in 3 forms. The first form is the free form, depicted by E, which is the low activity conformation that Sierra mentioned earlier. It's the activated protein C anticoagulant pathway. The bound form, E:(Na), the high activity conformation, and that's the conformation that cleaves procoagulant substrates more rapidly. And then the equilibrium form, E*, in which sodium is not able to bind thrombin at all. In this manner, sodium acts as an allosteric regulator for the activity of thrombin and balances out the blood clotting process. This is just another diagram of the protein itself with the exosite 2, the exosite 1, and the active site in the middle. The thrombin amino acid sequence consists of 622 amino acid residues listed in order 1 by 1 by letter code below. If you would like to write them all out, have at it. We decided not to. And so now I want to talk about thrombin at the gene level. It is coded by the F2 gene, or coagulation factor 2. It is located on the short arm of chromosome 11 at position 11.2. And here's a nice little graphic for you to see that. Mutations can occur, like in any gene, in the F2 gene, and the main 2 causes these mutations create are prothrombin deficiency or prothrombin thrombophilia, so too little prothrombin or too much prothrombin. The deficiency is caused by 50 plus different mutations that exist, and they usually change one singular amino acid, which can lead to a varying decrease or increase in protein activity. That leads to bleeding episodes of varying severity. So you can bleed out or you cannot notice that you have a mutation at all. On the other side of that, you have thrombophilia, which is a mutation that changes one DNA nucleotide, guanine, to adenine at position 20210. This leads to an overactive form of the F2 gene, and it creates too much prothrombin, which in turn creates excess clotting, so unnecessary clotting, which can cut off oxygen flow to limbs, extremities, organs, tissues, what have you. Can also result in pulmonary embolism, which is not a fun thing to endure. More on the gene level, there are 9 different mRNA sequences that exist for the F2 gene. There are varying degrees of protein function, some more functional than others. The mRNA exon sequence for the F2 gene that produces 622 amino acids is illustrated below.
So using the sequence that was just on the screen, you can create a primer sequence for qPCR, which is just a way of experimentally and in the lab amplifying or like overexpressing a gene of interest. So you can create the forward and the reverse complement primers so that when you put them into your qPCR mix with your DNA, you can kind of doubly sequence or make that little region of the gene. So for the forward one, we went ahead and used the first 85 base pairs, and then with the reverse, we used the final 95, just so then you have like kind of a sticky end of in between of where you want to replicate that during qPCR. The reverse complement, because of the double helix nature and the 3 to 5 prime anti-parallel nature of DNA, requires you to flip your forward primer sequence and then fix those base pairings again, so then you have that complement structure for your primer.
So now we're going to segue into the primary source journal article that we chose. It is called The Role of Thrombin Exocytes 1 and 2 in Activation of Human Coagulation Factor 5.
So the primary goal of this experiment was to study the thrombin's exocytes 1 and 2 on their specificity of cleavage sites on factor 5. So as I said before, the coagulation cascade is a very complicated series of intrinsic and extrinsic pathways that mediate the formation of fibrin in that clotting process. Currently, there are 13 known coagulation factors. In this study, they specifically looked at factor 5. So to test everything in this experiment, they needed to be able to determine the protein concentration. They had to create recombinant factor 5s, and then they had to be able to measure those expression levels. So to determine their protein concentration levels, they used micro-BCA protein assay with BSA as a standard. To further identify the concentration of thrombin, they used chronogenic substrate S2238, which is just a ligand for the thrombin active site, which is then used in the BCA assay to further quantify the concentration. For the factor 5 activity, they measured its ability to stimulate factor 10, which is just another procoagulation factor in that thrombin activation cascade. And in order to avoid the negative feedback loops of factor 5, they used a reversible thrombin inhibitor during the assay just for that negative inhibition. To create those variants of factor 5s in order to test the different specificities of exocyte 1 and 2, they created variants of factor 5, which is pretty much just a one residue mutation in factor 5 to see if that affected the ability of thrombin to cleave at that site. So they created 6 different variants using plasmid DNA and transfected those into COS1 cells for testing. The wild type strain was arginine 709, arginine 1018, and arginine 1545, which will become a lot more specific later on. Once the reactions occurred, they were able to estimate the expression levels of factor V using the ELISA assays. The primary antibodies they used were 3B1 and the secondary was HV1. And they did this in order to determine the activation rate of factor V by thrombin using a steady state rate determination. So because there are 2 exocytes on thrombin, they had to kind of eliminate one during testing and then eliminate the other for different series of testings. So for exocyte 1, they used, or to block exocyte 1, they used something called an aptamer, which is pretty much just kind of like an inhibitor, but for the exocyte rather than the active site. So aptamer 1 would block exocyte 1, which allowed them to test exocyte 2's specificity, while aptamer 2 blocked exocyte 2 and therefore allowed for the specificity of exocyte 1 to be measured. To kind of conclude their experiments, because they had some results that kind of conflicted each other, they used an SDS page during the reaction time to see if at a certain point the factor V kind of reached its maximum activity. So they did so by collecting samples during a variation of an hour, and they placed those on a gel just to examine the factor V's like amount at varying time spots.
So the results, the exocytes 1 plays a critical role in the recognition of arginine 709 cleavage site, and that was backed up by an SDS page analysis in which arginine 709 cleavage and formation of factor V heavy chain were significantly delayed in the presence of aptamer 1 but not aptamer 2, indicating that exocyte 1 is important in the formation of that heavy chain and arginine 709. Exocyte 2 is involved in the cleavage at arginine 1545, but not arginine 709 or arginine 1018. And that was backed up by another SDS page analysis in which cleavage of arginine 1545 and formation of factor V light chains were significantly delayed in presence of aptamer 2, indicating that exocyte 2 had a role in the cleavage of arginine 1545 and formation of light chains.
So figure 1 is the activation of factor V cleavage sites by thrombin. So this was kind of like their initial test just to get like an idea of what they're working with. So each of these lines depicts the different variants that they created for factor V. The key importance for this first trial was that this upper line right here denotes their wild type factor V, and just below it, which means it pretty much has the same activation or activity of factor V, is the RIR variant. This suggested to them that compared to their other variants like QIQ, that the RIR variant showed that exocyte 1 is pretty specific for that arginine 709, which then led them into their following experiments.
So these 3 graphs are of a few different variants. It's over a period of time. It's the FV cleavage site mutants in the presence of thrombin exocyte aptamers 1 and 2. You can see in the RIQ graph you have a really low factor V percentage when aptamer 1 is present, suggesting that exocyte 2 is probably involved in arginine 1545 recognition and cleavage. And then over here in the QIR plot, you can see that aptamer 1 and 2 did some light or actually it's pretty heavy inhibiting in those bottom 2 lines, and then at the very bottom where both aptamers were present, you got virtually no activity on the enzyme at all. And this was kind of confusing to the researchers when they first got these results back, because they were kind of stuck. They were like, okay, well, what does exocyte 2 do? Because, you know, we have these results and we're not really sure. They're not giving us a solid conclusion on what exocyte 2 does. So that's when they decided to run the SDS pages, and that's where they really made their conclusion that exocyte 2 played a role in arginine 1545. If you look really closely here, you can see the light chain is what's associated with cleavage of arginine 1545, that formation. So here, where we're inhibiting exocyte 2, the light chain doesn't form till around 25 or 30 minutes. And when we're not inhibiting exocyte 2, the light chain forms down here around 10-15 minutes. which indicates that exosite 2 definitely has something to do with light chain formation and the cleavage of arginine 1545.
So after all these tests, since they had those kind of conflicting results at first, like Tara just explained, after running the SGS page, they kind of came to the conclusion that their major results were that exosite 1 plays a role in the cleavage of arginine 709 on factor 5, while exosite 2 plays a major role in the cleavage of arginine 1545 instead of 709 or 1018. So that was kind of their major results after having that bit of confusion. And that's our presentation.
Yep.